today I'll be talking about linking hairy vetch germplasm diversity to traits that may facilitate improved nitrogen fixation. And I'm going to show a lot of data today, which is kind of an experiment on you guys. So what I did was I put a lot of data in the talk that you may not always come across in terms of understanding how to interpret it, but it's really pretty easy. And as you can see in this slide here, we have a gradient. On this side of the slide, we have some hairy vetch. You can't see it that well, I guess, that had winter killed, and you have various stages of vegetative growth, and then you have some flowering genotypes down here. This picture was taken on the same day. These accessions were all planted on the same day, the previous fall, and this picture was taken on May 15th, and you can see that variation, that gradient. So that pattern, and that's what I want you guys to do with the data that I'm going to present, is just look for patterns. So that's all. And I try to point them out. I try to draw boxes around them, things like that. So don't read any numbers or anything like that. Patterns and labels, and then everything will be fine. But this gradient points out a range of diversity that we see within the vetch germplasm, and I will tell a little bit of the story here. So why vetch? Well, vetch is used in sustainable ag systems around the world. Some of the positive points, so winter annual legume, high biomass and nitrogen fixation, cold tolerant, evidence of disease suppression, although it was way up on your chart. We've seen suppression of fusarium in watermelon systems. Improved soil aggregation and weed suppression. Some of the drawbacks, there's a lot of reports of some hard-seededness in these varieties. It's maybe an applicant outcrosser, self-incompatible. We're not sure about that one. It's susceptible to extreme heat and cold, and it may harbor nematodes. So there's some issues with it that we have to overcome. And this was in there to remind me that there are only a few improved varieties. AU Early Cover, Roth was developed by a farmer in Pennsylvania. Purple Bounty and Purple Prosperity were developed at the USDA. Okay, so here's how vetch might fit into a typical small grain system in Beltsville, or on the East Coast for that matter. I have a little diagram here that points out this kind of cartoon that I try to use to represent rotations. So you have a time frame of planting, a time frame of maximal growth, and then a time frame of management action. So in year one, you might have hairy vetch preceding corn, and that's going to provide your nitrogen for the corn. And then you'll go into a winter rye. Year two, the winter rye gets maybe rolled or whatever your practice is into soybean, then winter wheat. And then maybe years 3 through 6 have perennial wheat or alfalfa. I was glad to see the perennial wheat varieties in your last talk there. And so although vetch is maybe one part of this whole rotation, from a system standpoint, we think we have to optimize everything that we're planting in the field. So we have to pay attention to cover crops just as much as we pay attention to our cash crops. Because if we don't optimize every part of the system, we're just doing ourselves a disservice in the sense that we're expecting this cover crop to perform well within this highly tuned system. But if we don't focus in and optimize each one, the system may have faults. So although hairy vetch plays maybe a small role in this rotation, we're trying to optimize it. Here's some images of us making some raised beds and tilling in vetch for watermelon production on raised beds. The corner picture is Julie Grossman's team down in North Carolina rolling vetch, and then they're going to no-till corn into it. And then on the right here is just a plug for our long-term experiment in which we use vetch in the rotation that rotation I showed you earlier. It's a farming systems project in Beltsville. It's been in place for 16 years, and it's a comparison of 5 systems. 2 of them are conventional, 3 of them are organic, and we grow corn, soybean, and wheat. And it's one of only 3 long-term sites in the country where you can actually do a direct comparison between organic and conventional grain production on similar soil types. So if you're in Beltsville, come and visit us, or if you want to collaborate on this project, let me know. I have my contact information up here. So there's a lot to do in that project. So here, there's a few improved varieties out there. Tom Devine was my predecessor at the USDA. He was plant breeder, or is plant breeder in, he did a lot of alfalfa, soybeans, some birds of trefoil, and he was in Beltsville working on Vetch. He used some of the early lines, AU early cover, and some of the close relatives to create Purple Bounty and Purple Prosperity, which flowered earlier in the season, which was an attribute that some of our farmers wanted, especially the ones trying to practice no-till organic. They can control the vetch once it has flowered, so they'll roll it and it'll stay dead. Purple Bounty, Purple Prosperity have some coal hardiness that AU early cover doesn't have. They're not great, but they work better, and they produce a lot of biomass. He recently retired, and I inherited this project, and so this is kind of a little story in itself. I'm sitting in my office about a month into getting my job at the USDA, and my boss walks in and says, "Hey, Tom Devine's retiring, and he's got that field full of vetch. Can you do anything with it?" And I'm thinking, I better do something with that to make a good impression. So the experiment that I inherited was this a field in which Tom had planted out all 64 accessions that were collected from around the world that were deposited in the USDA Germplasm Collection. And these stars here are the locations of where they were collected. And then the table at the bottom is just a table of where those different accessions are from. And by training, I'm not a breeder at all. I'm not an agronomist. I'm a molecular biologist. And so I said, well, what do plant breeders do when they want to look at the germplasm collection? And I found this paper by Steiner et al., where they took birds of trefoil germplasm collection, and they just characterized it. And then they looked at how related each of the accessions were, so they could have an idea when they wanted to go into a breeding program what they were starting with, really. And so I looked around for one for vetch, and there wasn't anything there. So I said, oh, maybe this is what I should do. So the goal was set to characterize the germplasm collection and understand how related each of these accessions are as a starting point for you guys to use that material to then start breeding. And what we would like to do is understand how related all the accessions are, and then start assigning traits of interest like early flowering, late flowering, cold hardy, susceptible to cold, and anything else that the farmers come to us and request some traits that might be available. We could then overlay them on this phylogenetic map. Yeah?
Relating back to that map, they're wondering where the germplasm center is for area that.
The germplasm center, I believe, is in Pullman, Washington. And if you go to the USDA Germplasm Collection or the GRIN database, you can make requests for any of these accessions that I will talk about here. Excuse me? Oh, the center of diversity. Like the origin is thought to be in Turkey, in this area. Turkey and Iran. Area right there. So that's where it's thought to be. I can comment on that afterwards about trying to analyze that. We did a little work on that, but I'll just keep going with this. So I looked in the literature. I didn't see anything for vetch, so I said I'll use my skill set, molecular biology, to go in and conduct an experiment or an analysis, really a survey. We used this AFLP analysis, which is actually has a big name, but it's actually very simple. The goal was to characterize the genetic relatedness among all vetch accessions. And AFLP is basically this. We go and collect biomass. So that's Vetch. We collect it, and we extract the DNA from that. We cut it up, not with scissors, but with enzymes, and then separate that genomic DNA based on size. And I'm not going to go into the details of how we do it, but here's where we look at our first pattern, right? So these are the different sizes of the genomic DNA that we got from our Vetch accessions, 6 accessions here. And if we look at this pattern in A and B, we can tell A and B are the same, right? And C and D are the same, right? And then if we look at the top band of this and the bottom band, we have 4 bands that are in common between A and B, and these 4 bands that are different. So we could say A and B and C are related, but they're not identical. And this is the same thing that they do. You ever see one of those TV mystery show or what do they call it? The CSI and things, things like that. When they do all that funky stuff in the lab, this is what they're doing. And then we look at E and F, and every band in E and F is different from every other band. So we know that E and F is not related to that. So that we use this pattern to say how similar are they to each other and how different are they from each other. And this is the same thing that you would do if you were doing a genealogical study with you. You would have some bands that were common to your mother and some bands that were common to your father, but there would be differences between them. And you could go back through your lineage and you would see some bands that were common on one side of your family, some that were other, and you would be the unique result of all those combinations. So this is how we can represent those similarities and differences. And this is basically a genealogical tree in some ways. These are all the accessions that we've looked at, and the accessions that are close together to each other are more similar to each other than those that are far apart. And I've colored them, which I guess you can't see that well. But what you can see is we have down here purple bounty, purple prosperity, AU early cover, all these accessions that we have worked with. Those are the ones that we've tried to improve. And then these different clusters, there's Albert Lee, variety not stated from Albert Lee Seed House, and then this one up here, Nebraska Common, which is probably a derivative of Madison, which was developed out in Nebraska. Most of the diversity that we're working with in terms of plant improvement within the Vetch is all in this one section down here. So all of that diversity we're really not using. And so that was interesting. I thought, hey, first of all, it kind of, I did this blindly, and the fact that the accessions that we know are relatives of each other clustered together was kind of proof of concept, which I was really happy to see. But then also what I thought was interesting was that there's such a wide diversity that's out there that we're really not using for anything. So along with this study, while the plants were growing out in the field, I thought, I better have some other information to connect to these to this genetic information, because it's really kind of useless to just have a phylogenetic tree if you don't know what the characteristics of those plants are. So we did things like track flowering date and nitrogen uptake and biological nitrogen fixation. We did a little bit of root work. And what we were able to do was this, we assigned these clusters to the different groups. And what we were able to do with our trait data was indicate whether one of these accessions was early flowering or late flowering. And the dark circles here are green, and they indicate the earliest flowering accessions. The red circles are, well, they're red, and indicate the latest flowering accessions. And when you look, the pattern here to look for is that most of the green circles are down in this bottom section. of the tree. And most of the red circles are up in the upper section. And really, it's the tree kind of bifurcates into two groups, one that's mostly early flowering and one that's mostly late flowering. And I thought, wow, that's great. That's what at every field day farmers come up to me and say, we need improved vetch varieties. I want one that flowers early and flowers right on this date. I said, okay, we could cut the germplasm in half and just look at these ones that are early flowering if we want to do some more specific studies. So that was great. I did all this information is in a paper in Crop Science, and I have the reference at the end, so you can go back and you can read the details if you're interested. So then I started a collaboration with Julie Grossman at North Carolina State and Napi Matapo, who's on the left here in that picture. Julie's on the right. She's a Fulbright scholar from South Africa, and we wanted to explore rhizobium ecology as it relates to the host genotype, right? So how do these different accessions of vetch that are all related in some way, do they have any impact on the rhizobium that are occupying the nodule? That's a new site to occupy, right? Anyway, so what we did was we got together with this map, this phylogenetic tree, and we picked pairs of accessions that were related to each other. And I mean, they're all related to each other, but these sets, these pairs, come from right next to each other on the tree. So there's two from up here, two from here, two from here, two from here, and two from down there. And so these 5 groups, and we wanted to do a number of different types of analyses of rhizobium function. So we looked at hairy vetch biomass by nodule mass, and we see this great relationship here. And I don't know which one should be the dependent variable in this case, but as biomass increases, nodule mass increases. So there's a positive relationship here. If you look at nodule number and biomass, you see that positive relationship here. So it looks like and this is across all these accessions, it looks like there's a relationship between the activity and the function of the nodules and the plant biomass, which is kind of 101, right? We all know that, but this is nice to see empirical values showing that. And we also looked at nodulation. I guess we could call it efficiency. On the y-axis there we have a number of nodules, and then on the x-axis we have a number of these different accessions, and they're grouped in their pairs. So we see that there are there's some differences between. So there's another pattern that we can look at that we have one case with the varieties from Turkey that are low nodulating, both of them, and then we have case like say this group 2 here from Iran and Nebraska. We have maybe one high, one low. So if we look at it, well, these colors aren't great, but these red ones we're considering low nodulating, and the green ones are considering high nodulating. And to complete this objective of trying to understand the relationship between the genotype and the rhizobial population or community, we can use some of the same tools that we use to understand the genetics of the vetch. And so we examined the rhizobial population size and structure using this technique called box PCR, which we don't have to worry about the details. And that gives us the idea of who and how many rhizobium are there in the nodules. And so we have a very similar diagram to before, but in this case, instead of collecting vetch biomass, we're collecting nodules from all these different accessions that are being grown in soil. We extract the DNA, we cut that DNA up, and then we do the same comparison of the sizes of the DNA and do that same sort of analysis where we say those that are most similar to each other are going to be clustered together in our results, and those that are most dissimilar will be separate from each other. So this what we have here is it's basically the same thing as that phylogenetic tree that I showed you before, except that it's in, as you see, it's exploded. It's in multiple directions. And what this is a graph that shows the influence of the genotype on the diversity of the rhizobial population within the nodules. And what we can imagine this as the center of this graph is the average diversity among all of the rhizobium that are across our whole experiment, right? And those arrows that are pointing in the same direction and close to each other, the rhizobium in their nodules are much more similar to each other than the arrows that may be pointing in opposite directions. So, for example, the Afghan 1 and purple bounty have fairly similar populations of rhizobium in their nodules. They're very different than the turkey 1. Okay? And the same goes for this turkey 2 and purple prosperity. Those they're pointing in opposite directions. They're very different. Those that are pointing close to each other are very close. So we use this analysis because we can look at the whole experiment in one picture. And I like doing this not to quantify things, but to look for patterns and to try to understand. Well, we may not be able to say that X is greater than Y, but we may be able to say that X and Y are close together in the alphabet, kind of like that. And we can use this to build hypotheses to then test. And when I looked at this, I had the advantage of being able to look through all my data in front of me, not like you guys where that slide is gone. I was thinking about this slide here, where we had low nodulating and high nodulating. And if we look at this graph and think about that slide, we see that all the accessions that are low nodulating are on this side of the graph, and all those that are on high nodulating are on that side of the graph. Well, that's great. That gives us some. So what this is telling us is that across all the across the whole diversity of the germplasm, there seems to be some differential between the rhizobium that occupy 5 minutes. Good, almost done. That occupy the nodules of these low nodulating accessions, and the rhizobial community in these high nodulating accessions, they're different from each other. And that, to me, says that the plants are having some control on this. There's some interaction here. And if we look at these values on the X and Y axis, we can calculate how much impact the genotype is having on the structure of that microbial community. And if you add those both up, it's about 45%. So about 45% of the diversity, the difference in rhizobial community is driven by the genotype. About 45% is driven by the Vetch genotype. So I also saw this other pattern where going left to right, the late flowering accessions are on this side of the graph, and the early flowering are on that side of the graph. So that's another thing. Okay, well, maybe there's some interaction between early flowering, late flowering, high nodulating, low nodulating. And now this is where I'm going to get really crazy on you. We can overlay them, and the reason I left this in, I thought this would be way too confusing to show, and then I thought, no, it's not going to be, because my neighbor, who always comes to see my talks and stuff, he said, oh, what you got to do is put a Venn diagram in there. Everyone loves Venn diagrams. So this is a Venn diagram, basically. And what we can do, looking at these overlaid, these images, we have an area that's high nodulating and early flowering. This accession is low nodulating and kind of intermediate in flowering. And we have late flowering and low nodulating over here. And then we have these ones down here that are high nodulating and late flowering. And to me, this gives us the ability to then say, okay, what are the traits we're interested in? Maybe we pick from this group for one thing, we pick from this group for another thing, and we can use that as the basis to go forward. So if we go back to the phylogenetic tree, and we overlay our high nodulating and low nodulating, we see that that's dispersed throughout the tree. We have some clusters where we have both high nodulating down there in the bottom and low nodulating at the top. And we can do this is basically one way to go forward. We can do other things with this collection, like look at biological nitrogen fixation, and the column B on the right is just the percent of nitrogen derived from biologically fixed nitrogen when these were grown in the field. And these are just means of the group, so we don't know how consistent this would be from year to year, but this could be a basis for going forward to focus in on that trait as well and try to make connections between the nodulation efficiency and the amount of biological nitrogen fixation. So in conclusion, Vetch genomic markers were able to predict relationships between known related provenance of Vetch accessions. So the tree worked. The arrangement of those seems to have some basis in reality. There's predictable patterns among genotype and flowering. Nodulation and nodule mass is correlated to plant biomass and nitrogen accumulation. And the Vetch genotype was responsible for about 44% of the rhizobial community structure. So some questions headed forward, and these are kind of like questions that if someone has an answer to or ideas of how to go about this, I'm all ears. Hard seededness, Vetch and mixtures, we're working on this now, looking at the impact of competition on biological nitrogen fixation, lead suppression, and decomposition. Cold hardiness, or the question of adaptation to climate change, is an issue that we're interested in. And we're doing some more explicit work on flowering mechanism and outcrossing. Flowering mechanism, trying to understand how Vetch actually transitions from vegetative to flowering, and whether or not we can use early indicators to select for early flowering. And that's it. Thanks to these people, of course, in lab. And that reference at the bottom is the paper that I referred to for the earlier parts of this work. So.